All right, let's go ahead and get started. Um, so before we jump into the content for today, a couple quick announcements. We have the methods assignment that's due this evening. Again, that's meant to just be a couple of paragraphs that are meant to help provide you an opportunity to think through how you plan to test your hypothesis. This is for completion. It's meant to help keep you on track and give you an opportunity to get feedback on the suitability of that approach to test your hypothesis. And so put forth your best effort, but then also it's an opportunity for you to get feedback to make sure you're employing the best methods or approaches to test the hypothesis that you've written. And again, I'll repeat it multiple times throughout the course of our semester together. If you need to pivot your hypothesis or alter it or change it, again, that's okay. That's literally something I was talking about with one of my grad students for their PhD today, where it's okay to need to alter or pivot or improve or refine your hypothesis. And so if you find that to be the case, that's totally fine. That is a natural part of science. The second announcement is our peer review of the 3-minute theses is going to be due on Thursday. You all should have gotten by now a Google folder, access to a Google folder with the other videos that you'll be providing feedback on. It should be 4 of you in a group, so you'll be providing feedback on 3 videos, and there's a feedback form to help guide the feedback that you're offering. And we want it to be both concise and also incisive, meaningful feedback. And so try and be as constructive and helpful as possible. Is there anyone that hasn't gotten a Google folder sent to them yet? Great. Just want to make sure. Um, okay. With that being said, we can go ahead and jump into our content for today. So we discussed DNA replication over the course of the last 2 lectures. Today, we're going to be jumping into transcription. And our next lecture will be talking about transcriptional regulation. But today, we're going to be talking about the foundational pieces that make up transcription. And we'll be talking about that in the context of diabetes. Diabetes mellitus, or diabetes as it's colloquially known as, it's actually a group of metabolic diseases. And we say group because there's multiple different types of diabetes. But each of them are characterized by hyperglycemia, which is elevated blood sugar levels that results from some sort of defect either in the secretion of insulin, insulin action, or the responsiveness to insulin, or it could be a combination of both. And so if you're asking yourself, like, why is this important? Why is insulin so important to this process? And that's largely because when you consume a meal or consume some sort of beverage that has sugar in it, what ends up happening is that that sugar is taken up across the epithelium in your small intestine. So you get that blood sugar going into your bloodstream that then is accessible to your cells throughout your body as an energy source. And there's transporters on the membrane of all of our cells. They're glucose transporters. They're usually called GLUT and then a number. The most common one is called GLUT4. And so the ability of our cells to take up this glucose is mediated by insulin, because once that glucose gets into your bloodstream, it triggers insulin from beta cells in your pancreas to be secreted. Once insulin binds to insulin receptors on our cells, that increases glucose transporter expression, GLUT4 expression. And so when insulin binds to its receptor, we get more GLUT4 expression, and that makes it easier for glucose to leave our bloodstream and go into the cells and be used as an energy substrate primarily in glycolysis. And so Unit 4 is going to be all about metabolism and what glycolysis is. And so that ability for insulin to be released by the beta cells, or the ability for the insulin receptors to bind to insulin and enable glucose to be transported in, is critical for our basic or fundamental cellular metabolism. And so what ends up happening is that the chronic hyperglycemia that results from diabetes, where glucose can't be transported in, has a lot of complications that are associated with that hyperglycemia. When we have this buildup of blood sugar that happens over a sustained long period of time, it's going to create a lot of dysfunctions within the small blood vessels in your body called capillaries. And so one primary example of that is going to be our eyes. For diabetic patients, it's going to be a dysfunction in the eyes called diabetic retinopathy. It's going to create dysfunctions within those capillaries in the retina that can lead to blindness. There's diabetic nephritis. There's going to be issues in the kidney where the capillaries present within your kidneys down to the nephron units are going to be clogged and won't be able to sufficiently filter out contents from your blood. And so there's going to be kidney damage that can eventually lead to either needing dialysis or having kidney failure for diabetic patients. There's also diabetic neuropathy. There's going to be nerve damage that ends up resulting in loss of sensation in their extremities. There's lots of instances where patients with chronic hyperglycemia have such severe diabetic neuropathy that they end up developing these really painful lesions and ulcers on their feet that eventually lead to either a toe or whole foot amputation. There's going to be a series of complications or dysfunctions that result from these capillaries being altered as a result of this chronic hyperglycemia that lead also to cardiovascular disease and some sort of coronary artery disease as well. Some of the symptoms that are associated with diabetes, polyuria, that's frequent urine, polydipsia, constant thirst. There's a lot of weight cycling or severe weight loss, not cycling, forgive me, severe weight loss that can occur. There's going to be impaired growth, and this is particularly in the case with younger patients, typically below 13 years of age, where it's going to be harder to see that impaired growth in an adult patient. But for those who are 7 to 13, that impaired growth is going to be a little bit more noticeable. And finally, one of the final complications that can be really problematic is ketoacidosis. So what ends up happening is that when the sugar that's typically taken up by cells and used as an energy substrate, if it's staying in the bloodstream, it can't be used as an energy substrate by cells. What ends up happening is that lipids become a new alternative energy source for our cells. And as lipids in our free or particularly fatty acids are used as an alternative energy source, some of you might think that it sounds like the ketogenic diet or being in a state of ketosis, which is somewhat, there's a range of where that's acceptable. That range being how much ketone bodies accumulate in your tissue. And the ketone bodies are what are a byproduct of breaking down fatty acids as a new energy source. But what ends up happening is that these ketone bodies are incredibly acidic. And so particularly for diabetic patients, excuse me, when these ketone bodies build up within the bloodstream or tissues, it becomes incredibly acidic and becomes incredibly problematic and can result in being in a state no longer in ketosis, but now ketoacidosis, where there's a dangerously high level of acidity in the blood or in the tissue. Long-term complications of diabetes, again, includes retinopathy, diabetic neuropathy, because we also have these small blood vessels, the capillaries, all throughout our brain. There can also be cerebrovascular complications that emerge as well. The changes to the blood vessels can also lead to their stiffening, which leads to hypertension. There's going to be, again, alterations in lipoprotein metabolism because we're breaking down lipids as a new energy source here. And there's also instances of periodontal disease. So with the increase in blood sugar, there's going to be classes of bacteria that exist in the gums that colonize within the gums that can become incredibly problematic and lead to infection and periodontal disease. With the polyuria and polydipsia, what ends up happening is that with this increased blood sugar, what ends up happening there is that there's this osmotic pressure as there's the high sugar content that's being filtered out in the kidney is going to be driving an incredibly high osmotic pressure for more water to be filtering out of your kidneys and into your urine. And so that osmotic pressure that occurs explains the incredible amounts of polyuria that happens among diabetic patients who are kind of in the earlier stages of their disease, who haven't started monitoring their blood sugar quite yet. And as you can imagine, with increased rates of polyuria, increased rates of urination, there's going to be a a far more, there's going to be an excessive amount of thirst that is associated with that. What's important to note is that, we now have a variety of measures that help us with diagnosis and with trying to monitor and treat diabetes. Some of the earlier diagnostic approaches were a little bit more rudimentary. So I just explained how the polyuria, there's a lot of that sugar that's being filtered out into the urine. One of the earlier diagnostic approaches, early 20th century, for determining whether or not someone had diabetes was to either sniff or taste their urine to see if there was an excessive amount of sugar in their urine. Thankfully, we have blood sugar monitors now, which doesn't require that. But that's just meant to try and illustrate the extent to which how much sugar can actually be excreted out among these diabetic patients. As you can imagine, there is a high total cost, high economic impact associated with diabetes. Total cost, which means direct and indirect expenditures, as of 2017 was 327 billion. I suspect by now, as our variety of metabolic diseases that are associated with diabetes continues to increase, as well as an aging population where there reaches a point where if you reach a certain age, most patients become diabetic. So the total cost for this by now probably has exceeded 500 billion, I suspect. Incidence rate is incredibly high with about 1.9 million cases per year in the US. There's an interesting demographics that are associated with the prevalence of diabetes. It's typically rare, more rare in African or Asian populations, and some of that can be attributed to their diets that are very different than the Western diet. It varies by race across the U.S. population from 7 to 14%, with approximately an 8.3 average overall. There are some populations that have incredibly higher rates of diabetes, that being the Pima and Papago Native American tribes, but that's not a type 2 diabetes. It's more so of a genetic defect that predisposes that population to have a much higher rate of diabetes. And again, it's worth noting that we have 3 different kinds of diabetes. We have type 1, which is an autoimmune form of diabetes, where the autoantigen are the pancreatic beta cells that secrete insulin. Type 2 diabetes, which is an insulin resistance, and then type 3, I'm actually skipping myself here. Type 1, which is the autoimmune form that sees beta cells from the pancreas that secrete insulin as the autoantigen, as the foreign pathogen that's attacked. Type 2, which is much more common in the United States, is associated typically with improper regulation of either insulin or insulin signaling. That's oftentimes colloquially referred to as insulin resistance. Another example is called mature onset diabetes in the young. And finally, a third form is gestational diabetes, where pregnant women develop diabetes during pregnancy, but then usually it goes away. What can happen is with some patients is that if they have multiple bouts of gestational diabetes with each pregnancy, they can eventually develop type 2 diabetes for the remainder of their life. And so we'll have this be our jump off point to jump into transcription. But before we do so, I'll stop for any questions on diabetes. Yeah. Sorry, I should have been more clear. There is something on the autoantigen will be a protein that's specific to pancreatic beta cells on their surface. Not the entire cell itself, but rather each group of cells usually have a unique marker or protein on their surface that's specific to them. And so for type 1 diabetes, there's a protein that's specific to beta cells that's seen as the autoantigen that unfortunately results in the beta cell destruction and the inability to produce insulin anymore. Yeah. Not that I know of. Yeah, I mean, with each of these, with each of these, it's either no insulin, improper signaling of insulin, hormonal changes that alter insulin signaling, but none of it is there's no instance I can think of where there's an excess of insulin that leads to some sort of issue. But what's a good way to describe this? If there is excessive insulin, thinking of I'm just thinking of friends who are diabetic. Typically, and I'm sure a lot of you who have family or friends that have to deal with this, but typically if there's too much insulin being injected, that'll eventually lead to their blood sugar levels dropping dramatically, which can unfortunately lead them to being like in a diabetic coma. And so that's where typically will end up happening is they just need orange juice or some sort of sugar to try and get their blood sugar back to the right level. And so there's no disease I can think of where there's excessive insulin, but the consequence of excessive insulin for some of these patients is that blood sugar drops too low to a point where they might go into coma. Yes. Yeah, so hypoglycemic might be either a result of too much insulin or activity or doing some sort of activity level that that uses up the insulin in the blood sugar that to a degree that they weren't prepared for and can make them hypoglycemic. That can be dangerous. Yeah. Not that I know of, no. All right, so we're going to use this as an opportunity to switch and start talking about transcription. And so when we get back to transcription regulation, we're going to circle all of this back to diabetes, but we're going to try and hit some of the main points about transcription today. We're going to do a quick 30,000-foot view summary of transcription, and then we'll jump into each of the stages. And so again, as I try and highlight or point out for each lecture, like we're going to be hitting like the landmarks, the key points of transcription, and we'll go and provide a little bit more detail about each of these key steps. And so when we talk about transcription, we have a single transcriptional unit where transcription occurs. Within the eukaryotic system, that's going to be one single gene. In the prokaryotic system, there's going to be multiple genes within a single transcriptional unit. And when we talk about the regulation, it'll make a little bit more sense why there's multiple genes within one transcriptional unit in the prokaryotic system. But in the eukaryotic system, one transcriptional unit equates to one gene. And each of them have a few key pieces that make up this unit. We have a promoter where transcription begins, and we have a terminator where transcription ends. And these are sequences within the DNA itself. What ends up happening is that RNA polymerase will come in and bind at the promoter, and it will unwind the DNA at that site where RNA polymerase is. And as RNA polymerase begins to read the template strand, and it's going to be reading the template strand in the 3' to 5' direction, RNA polymerase is going to produce an mRNA in the 5' to 3' direction. So it's somewhat similar to replication with the DNA polymerase, where it reads the template strand 3' to 5', but it synthesizes the mRNA here in the 5' to 3' direction. And what ends up happening is that we have our template strand, and the other strand is typically referred to the coding strand. And so our RNA polymerase is reading the template strand, making an complementary and anti-parallel mRNA. And what's going to end up happening is that mRNA is going to match almost nearly identical to this coding strand. The only difference is that there's going to be a uracil there instead of a thymine in the RNA. And so it's going to continue to move down this transcriptional unit. And as it moves, it's going to continue to read the template strand, continue producing a longer and longer mRNA. The DNA behind it, where it once was, will wind back together. And elongation will continue until it reaches the terminator, where there's a termination signal, a cue for transcription to end. And once that happens, the mRNA will be released. RNA polymerase will unbind from the DNA, and transcription will have ended. And we have an mRNA transcript produced there at the end. For the promoter, the site at which transcription begins, again, it's reading the template strand, but the coding strand is going to end up matching the mRNA here. The site at which the first nucleotide is read during transcription is called the start point, right here. And so the promoter, that is kind of like the site at which the RNA polymerase binds, is just upstream to the left of that start point. And so what ends up happening is that this promoter provides a signal for where RNA polymerase binds, and just to the right of that promoter is where the first nucleotide is going to be read during transcription at that start point. And so these promoters are typically going to be possessing consensus sequences, these repeating units of nucleotides that are conserved across across species that serve a specific function. And here, one of the main consensus sequences for us to be mindful of is called the Tata box here within the promoter on the coding strand. And so that's going to be a critical the Tata box is a critical consensus sequence in transcription within the promoter of our DNA. And so if we look at the sequences across a variety of genes, but sorry, let me repeat that one more time. If we look at the nucleotides at the promoter across a variety of genes, there's going to be differences, but what is conserved across them is going to be these sequences here that includes on the right segment, the Tata box at the far right part of that right segment. So before we jump into this any further, I'll stop for any questions that you guys might have so far. Yes. Yeah, so what I should say is I'll take a step back. Consensus sequences at large are these conserved nucleotide sequences that serve a specific function. And so we talked about it in the context of replication with these origins of replication. Now, in the context of transcription, we're describing them as serving an important function within a promoter to signal the site at which RNA polymerase binds for transcription to begin. Thank you. Mm-hmm. All right, so for the initiation of transcription, we have, again, RNA polymerase binds to the promoter that possesses this Tata box. That's just to the left of the start point. And so once it begins, what ends up happening is that RNA polymerase will stay put for a bit, because what ends up happening is that we have these nucleoside triphosphates, or NTPs, that serve as kind of the building blocks for the mRNA nucleotides in this mRNA transcript. And what I should take a step back. I'm going to be using a couple words interchangeably. I'll refer to the mRNA that's produced during transcription as either mRNA, mRNA transcript, mRNA strand. If you hear transcript, think mRNA that's produced as a result of transcription. And so when the RNA polymerase binds at the promoter, and when we have our first nucleotides being added in to begin this mRNA transcript, what ends up happening is that RNA polymerase stays there until the first 9 base pairs have been added. And then once those first 9 base pairs have been added in on the mRNA, now RNA polymerase can start moving along. These 9 base pairs are basically kind of like the frame. You can think of it as like the size of the frame that is being added when initiation begins. But once those first 9 are added in, now we have RNA polymerase moving down the DNA. And you can almost think of it, this is kind of a reductive example, but when we think about the way in which the DNA is unwound in replication versus transcription, you can almost think of, when you're trying to look for something that you've lost in the couch, when you just kind of like stick your hand in the couch and like run it across, That's kind of like what RNA polymerase is doing. It's kind of like sticking in and running across the DNA, whereas DNA polymerase with replication, things are totally being melted. So basically imagine like ripping the cushions out, totally unwinding it and looking in. Whereas here with polymerase, it's sticking in, reading it there and moving across, and the DNA will wind back up right behind RNA polymerase. And so as it's continuing along, again, the DNA behind it will wind back up, and the continual addition of these nucleoside triphosphates, NTPs, is going to be continuing to elongate and grow this mRNA strand that is the main output of transcription. And so about 12 base pairs of RNA are bonding to DNA as it goes along. This is happening fairly quickly. Excuse me. And so, again, it's worth repeating that the mRNA transcript that's being produced here is elongating in the 5' to 3' direction. RNA polymerase is reading the template strand in the 3' to 5' direction, and the coding strand will almost be nearly identical to this mRNA because they will both be anti-parallel and complementary to our template strand. The only addition is thymine in the coding strand versus uracil in the mRNA transcript here. And finally, we have termination. So what ends up happening here is that there's going to be a series of nucleotides in the terminator sequence at the end of this transcriptional unit. In the case of eukaryotes, at the end of this gene. And it's going to encode for, it's going to possess a series of adenosines in the terminator sequence that then will encode for uracils in the mRNA. In addition to that, there's going to be a series of GC-rich regions that lead to the formation of this hairpin loop. Bless you. So the combination of this hairpin loop and the uracils at the end are going to, but even more so, this hairpin loop will serve as a cue for transcription to end. And so this hairpin loop forms again because it's a GC-rich region, and genes here are complementary to each other. And so there's going to be this self-complementary set of nucleotides that explain why this hairpin loop forms. And this hairpin loop, again, is going to be a cue or a signal for termination to end. So once that happens, we're going to end up having RNA polymerase unbinding from the DNA, the release of this mRNA transcript. And so that's going to lead to the end of transcription here. Before we move on any further, I'll give space for a couple of questions about these steps of transcription. Yes. How do you differentiate? Yes. Mm-hmm. That distinct, I mean, so what ends up happening is we know, okay, so we have sequenced the entire human genome. We know what mRNA encodes for some specific protein. So what we're able to do is to kind of like bioinformatically compare, and we know like where this gene is located now because we've mapped the whole genome. So we can see, okay, we know the mRNA sequence. We can go in and identify and see, okay, when RNA polymerase is reading this gene to produce this mRNA that produces protein, we can distinguish which is the template strand, which is the coding strand. How RNA polymerase is able to distinguish between the two, there's going to end up being the specific promoter sequence on one of those strands. And so it's going to provide the cue for RNA polymerase to bind there specifically, as opposed to randomly binding anywhere in the DNA. Does that help? Okay. Yes.
Rights for every promoter. like, bind multiple promoters?
Great question. So we have three different kinds of RNA polymerase, but the main one that we talk about in transcription is RNA polymerase 2. That is the polymerase that is responsible for producing mRNA. RNA pol 1 is for ribosomal RNA, and RNA pol 3 is for tRNA. But only RNA pol 2 produces mRNA. So what ends up happening is that we have this complex that associates with RNA pol 2 that dictates how readily it can. How far ahead am I jumping here? You know, I'm actually going to say, hold your question for 2 slides from now, or at least the next 2 slides will answer that. But to kind of like help cue it up, RNA pol 2 will be binding to all of these genes and mediating their transcription, but there are other factors that will, factors is the right word, factors that will influence what binds to what gene. But all of these genes have promoters, which is like a key signal for transcription to happen here. I'm going to go around this way. So go ahead, Ethan. Yeah. So it is the hairpin. That is the mRNA. What ends up happening is that within the terminator sequence of the DNA, it encodes for GC-rich mRNA to be produced, and those are complementary to one another. And so they'll end up, as it's being produced, it'll loop back on itself and create this hairpin. And so this hairpin serves as the cue or the signal to RNA polymerase 2 for transcription to come to an end, because it's encoded in that terminator sequence at the far end, the far end of the DNA. That is the again, it's the terminator sequence. It's the signal for transcription to end. And so it'll provide the cue and the nucleotides for this hairpin loop to form and for transcription to end. And again, it comes down to the nucleotide sequence. The terminator, I'm just going to speak louder for the sake of the recording, so I'm not yelling at you. The terminator encodes for a rich GC segment of the mRNA to be produced during transcription. When it's produced, this hairpin loop will form. RNA polymerase 2 will detect that hairpin loop, and that serves as a cue for transcription to end, for RNA polymerase 2 to unbind and to stop transcribing that gene, or that transcriptional unit, I should say.
Once it, I guess, it went to
Mm-hmm.
and back is here how would it, if it's very severe,
the terminator sequence is fairly large. So as it's being produced, it's still within that terminator region. And so once it's sensed while it's still in the terminator sequence, it'll unbind and stop transcribing that transcriptional unit. Yeah. Go ahead.
What's the difference in like how RNA polymerase happens, like in transcription versus translation?
Well, translation is producing protein. We haven't touched on that yet. But
Not translation.
Replication? So repeat your question one more time.
What's the difference? Like how does RNA polymerase know like what signal to know like what to do? Replication versus.
So RNA polymerase 2 is not involved in replication. We have those 2 kinds of DNA polymerases. Those DNA polymerases are involved in replication, and they each have their own function in producing the DNA. But the only RNA polymerase that we talked about was primase, that produces that RNA primer. Totally different flavor of RNA polymerase. These 3 RNA polymerases here are not involved with replication at all. And primarily, we're going to be talking about RNA pol 2 because that produces the mRNA that encodes for proteins. And so RNA pol 2, not involved in replication at all. Does that help? Great. Yes.
When you have those have the A to that's base pairs.
Yeah,
So since A and T are like the complement, and T are in the kind of use race binding to temporal strand instead of the equivalent strand going the opposite direction.
wait, sorry, say that one more time.
So what kind of like, how does the language that the race node binds to the like temporal strand on the TNF rather than putting strand going the opposite direction? Because like you have the A with the TNA retrieve the base pair is going to be A and T retrieve and go in the opposite direction.
That's the next slide. So we're going to talk about it in just a moment. So hold that thought. Any other questions? Yes. General. It's not specific. RNA polymerase 2 is not specific to any gene. RNA polymerase 2 is able to transcribe any and every gene within the genome. There are specific factors that we'll talk about in the next couple slides that are associated with specific genes that kind of aid in the function of RNA polymerase 2, but RNA polymerase 2 itself is equipped and able to bind to the promoter of any gene. All right, we'll go ahead and press on. So the promoter region within a given gene, again, we have our start point that's labeled +1 here. It's labeled as this like single black box. Around it, we have what's, when you see PY, that's a subclass of our nucleotides, pyrimidines. That's going to be cytosine and thymine, C and T. And so you'll have 2 pyrimidines, a cytosine, your start point, which is that adenosine there, and then 5 pyrimidines to the right of it. And so this is what's called the initiator sequence. This marks the start point for transcription. We have just upstream of that to the left, we have our Tata box. And then just to the left of that, we have what's called these B recognition elements. And so it's this combination. So like after this AT-rich, the Tata box here, we have this combination of different nucleotides, but have very specific combinations of them that work. That, again, this is on the coding strand. So these nucleotides that make up the promoter for a gene on the coding strand serve as the cue for RNA polymerase RNA pol 2 to come in and bind and read the template strand. And so there's going to be kind of this coordinated effort between, on a given gene, the coding strand and the template strand kind of working together for this gene to be transcribed. And so does that answer the question that you had a moment ago? Great. And so the combination of the Tata box and this initiator sequence, these pyrimidines around the start point, this initiator sequence plus the Tata box is called your core promoter. Your core promoter is going to be a feature. It's going to exist on every gene in the genome. There's going to be some initiator sequence in combination with the Tata box that makes up their core promoter. And so just upstream of that, you have your B recognition elements, and then just downstream from the initiator sequence, you have it's DPE. It's called the downstream promoter elements. But again, the key part is your core promoter. That's going to be the most critical part of your promoter. That is where RNA pol 2 is going to be coming in to bind to promote transcription. And so we have a class of proteins, and some of you may have come across this in your research for your projects, but there's a whole class of proteins that aid in the ability for transcription to occur. They're called transcription factors. They undergo some sort of activation event, typically phosphorylation, but not ubiquitously is it going to be phosphorylation for a transcription factor. But transcription factors will get activated somehow within the cytoplasm, translocate into the nucleus, and they will also bind to the Tata box of some specific gene and facilitate RNA pol 2 to bind as well to the core promoter. So this class of proteins, transcription factors, will facilitate or make it more easy for RNA Pol2 to bind to the promoter for transcription to occur. For specific genes, there are specific transcription factors that will bind to them. Some labs have dedicated their entire career studying specific transcription factors and what these transcription factors will bind to. The lab I did my PhD in, my advisor dedicated his entire career studying STAT5. That transcription factor was his entire universe. And so what transcription factors can bind to will vary from what kind of cell you're working in or what tissue you're in. But again, there's specific genes that these transcription factors will bind to at their top box that then will make it more easy for RNA Pol2. So the more that a transcription factor binds to the top box, the more readily RNA Pol2 will bind and promote transcription of that gene. And so there is a subclass or subgroup of transcription factors that are kind of unique. Most transcription factors need to be activated. Some do not. Those that do not need to be activated, we actually call basal transcription factors. And what's unique about them is that they are required to allow RNA polymerase to bind to the DNA. So at minimum, for transcription to occur on a gene, you need some basal transcription factor to bind that it's required for RNA Pol2 to come in and bind as well. Within the eukaryotic system, the main basal transcription factor is called TF2D. And so it will first bind to the TATA box itself. Once it is bound to the TATA box, now RNA Pol2 can come in and bind and transcription can commence. But again, as more transcription factors bind in addition to TF2D, whether it's my old advisor's favorite STAT5 or some other transcription factor you guys may have come across, the more of them that bind, the more quickly and more readily will RNA Pol2 bind and promote transcription. And so this combination of transcription factors with RNA Pol2 creates what we call the pre-initiation complex. And so as TF2D, sorry, let me say that differently because I may have misspoke. The combination of TF2D and these other transcription factors make up what we call the pre-initiation complex. So with that pre-initiation complex in place, then RNA Pol2 binds. So we have TF2D here at the TATA box. We have another transcription factor that comes in and binds as well, a second one that comes in. These make up this pre-initiation complex, and now RNA Pol2 comes in and binds at this core promoter, the combination of the TATA box and that initiator sequence. And so then we'll have transcription that occurs there. I'm going to stop there and give space for a couple of questions. Yes. TF2D is the basal transcription factor for eukaryotes. Yes. Yes, that's where it binds. Yep. Yes. Wait, I feel like you're asking like an ontological question. Are you saying it's almost like a chicken and the egg. If TF2D is a protein that's required for transcription, how did the first TF2D how was it made? I don't know. I will happily say to that one, I don't know. I'm sure there's that's the first time I've been asked that question. That was a good question. I'm sure I'm sure at some point, like during morphogenesis and during development, there's something that enables the first TF2D to be produced outside. No. Well, now it's just like it's a good question. I want to honor a good question with an answer, but I don't think I'm going to give you a good answer. So I'll leave it with I don't know. That's a great question. Yes.
And And then with the other transcription factors, it's like the initiation complex.
Mhm.
Do you need more than TF2B for RNA polymerase to bind, or could it just bind with that?
It could just bind with TF2D. It will more quickly bind with these other transcription factors, but at minimum, as long as you have TF2D, you'll have transcription occur. It would still be the pre-initiation complex, but it would just be TF2D itself. But I will say, typically in biology, there's usually going to be other transcription factors. Well, there's no March Madness to distract us today, you guys. It's just us and transcription. All right, we can press on. So when we have an mRNA transcript that's been produced at the end of transcription, there's a series of modifications that happen to it because for some of you that have worked in a research lab that may have done qPCR or that may have handled mRNA in some capacity, or whether you've done qPCR in Ideas Lab, you may have learned that mRNA is incredibly unstable, super easy to be degraded. It's easy for enzymes, RNases, enzymes that degrade RNA, that we have in the oil in our skin, in our sweat, just all throughout our body. And that's typically an immune defense mechanism from some sort of RNA-based pathogen. But usually in the context of research, it's typically frustrating because that makes it really easy for when you're trying to work with RNA, where if you're breathing over your tube, you've typically already degraded your sample. And so when mRNA is produced as a result of transcription, there's a series of modifications that happen that help stabilize it and protect it from degradation. Those three are, there's the addition of a 5' cap, there's the addition of a poly A tail, and finally, there's splicing, excuse me, splicing, which we've talked a little bit about already, but we'll provide a little bit of the mechanism for how that happens as far as what's known so far in biology. And so for the 5' cap, what ends up happening is that this is a backwards cap. And the reason why we call it a backwards cap is because when we look from one nucleotide to the next, there's a connection from the 5th carbon to the 3rd carbon, or from the 5' end to the 3' end of the adjacent nucleotide. What ends up happening here is that the 5' end of this methylated guanine is bound to the 5' end of the mRNA. And so we typically, because of that, because it's 5' to 5', we typically refer to it as a backwards cap. That again, it's a methylated guanine. It has a methyl group. And what ends up happening is that this is added during elongation. So as we have the first nucleotides that are added in during initiation, then as elongation commences and during the course of elongation, this 5' cap is added in. The function that this 5' cap serves is threefold. And so it first flags this mRNA for nuclear export. So after transcription is ended, this 5' cap tags it to be exported out of the nucleus and into the cytoplasm, where it can be used for translation to make the protein that it encodes for. This 5' cap also serves a critical role for protecting this mRNA from degradation by exoribonucleases. So we talked about exonucleases during replication, these enzymes that degrade nucleotide strands from the ends. And exoribonuclease is just one that targets RNA specifically. And so it protects it at one end, the 5' end, from degradation. And finally, it also marks it as a binding site for ribosomes, again, during translation. And so the 5' cap aids in moving it outside of the nucleus to enable translation to occur. It flags it for where ribosomes can bind, and it also protects it from degradation. This 5' cap is added on by what we colloquially refer to as a capping enzyme that only associates with RNA Pol2. What do you think that might mean if it only associates with RNA Pol2, this capping enzyme? Yes. Exactly right. It means that only mRNA is going to receive this 5' cap. The other it doesn't associate with the other species of RNA polymerase that are associated with ribosomal RNA and tRNA. The other modification is polyadenylation. What that means is that there's many adenosines added on to the end. So one end gets the 5' cap, the other end gets polyadenylated, or there's a poly A tail ended to the 3' end. And so usually what ends up happening is that because of how long our transcripts usually are, they're usually thousands of nucleotides long, on the order of, I'm gonna say on average, like 5 to 8 kilobase pairs, or 5 to 10 kilobase pairs long, so 5 to 10,000 base pairs long. Because of how long they are, there is a need to mark the end with important indicators for which end is which. And typically what ends up happening is that we have a poly A polymerase, or PAP, that finds this poly A signal that marks the end of where the important information is. And so what ends up happening is that this nucleotide sequence here kind of marks the end of this, marks the end of what is the critical encoding portions of this mRNA transcript. And so what ends up happening is that it's going to end up cleaving the transcript and adding in a poly A tail. The reason why the ability to add this poly A tail is important is because the 5' cap protects the 5' end. This poly A tail will be able to recruit in poly A binding proteins that protect the 3' end. And so, and it's also, the poly A tail serves an important function in translation. And so both the 5' cap and the poly A tail, the commonalities here is that they both protect the transcript from degradation and serve important functions that enable translation to occur, that enable this mRNA transcript to be used in translation to produce the protein that it encodes for. And so again, we have this poly A polymerase, PAP, that finds this poly A signal, this nucleotide AAU AAA, that marks the end of this important coding region of the mRNA. It's a site where it's cleaved, and then PAP adds in the poly A tail. The length of that poly A tail can differ from the prokaryotic to the eukaryotic system. It's about 20 to 30 adenosines long, whereas in the eukaryotic system, it's about 20 to 100 adenosines long. And if you ask yourself why adenosine? Why is that the nucleotide that's used as opposed to any of the other nucleotides? It kind of comes down to what's most readily available, I think, what's most efficient. So if we think about the substrates for energy, ATP, it's adenosine triphosphate, and there's lots of ATP or ADP floating throughout the cell, and its base is adenosine, what makes up this nucleotide. And so I suspect that it's best explained that using A as for the poly A tail is evolutionarily efficient based on what's readily available for an energy substrate. I'm going to do one more slide, and then I'm going to give space for a handful of questions. So to help try and illustrate this, we have our 5' cap here. That's marked by this G, which is the methylated guanine. We have our poly A tail with these poly A binding proteins. Both of these are protecting each end of the mRNA from exoribonucleases, which is marked as XRN here. And so exoribonucleases, exo meaning wants to work on the ends, wants to try and degrade this mRNA. And again, we need exoribonucleases. It serves an important function of trying to destroy RNA from that comes from RNA-based pathogens. But what ends up happening is that these modifications will only last for so long. So there's kind of a time limiting factor here of when an mRNA is produced to be able to use it to synthesize the protein it encodes for and for it to be used in translation. And so what ends up happening is that we actually have this whittling away slowly, where we have the slow loss of these poly-A binding proteins. In addition to that, we have a decapping protein that then releases off the 5' cap after that poly-A tail is gone. And once both ends are gone, then this mRNA is going to be fully degraded. And so one important thing you'll hear any scientist say sometimes is when you're looking at the expression of something, mRNA does not always equal protein. Just because transcription occurs doesn't mean you're going to get the protein. And so sometimes that's because this mRNA can be degraded. And so what ends up happening is that when a protein needs to be expressed, that mRNA needs to be used within some time domain because it will undergo degradation. I'll stop there and give space for a few questions. Yes. Plasmids, yes. Plasmids, we typically encode the poly-A tail in there. But at least what I was hearing you say was within our own DNA. Because there's a, so in terms of efficiency, I'll go back. This 6 base pair sequence provides the signal for the poly-A tail to be added in. So rather than having 20 to 30, or in the case of eukaryotes, 20 to 100 adenosines being added in, these 6 base pairs provide the cue for the poly-A tail to be added in. So in terms of space efficiency, this works much better than having the poly-A tail at least encoded in our DNA. Great question. Yes. Is there something that regulates the degradation of different mRNA? Yeah, that's a really good question. I will say I suspect there is. I don't know off the top of my head. I suspect there is. So, like, again, I'll share something that I think I've said maybe only once or twice. Bless you. This class is a class of half truths, meaning there are some things that we know that are fundamental molecular and cellular biology, but there are some things that we're still learning. And so, for example, like what regulates mRNA degradation and if that differs from gene to gene, I don't think we know that yet. I think we know some of the mechanisms, fundamental mechanisms behind it, but what that looks like from gene to gene, and perhaps there's a correlation or a relationship between dysfunction of that regulation and some specific disease. But I don't think we know that quite yet. That'll be my new cop-out answer when there was a really good question that I don't know the answer to. A class of half-truths. All right, we can go ahead and press on. So we have discussed how there are accommodations of exons and introns within a given gene. And that when we have the mRNA that's first produced, it possesses both the exons and introns. When the mRNA is first produced at the end of transcription, we typically refer to that mRNA transcript as the pre-mRNA. Once it undergoes processing, in this case, typically splicing, we'll have what we typically refer to as the mature mRNA that is then used for translation. And when we get our single polypeptide chain that the mRNA transcript encodes for, we've talked several times already about how there's different domains within a given polypeptide chain. We talked about subunits where there are multiple proteins that come together to form like a macromolecular structure. But within a single polypeptide chain, there are different domains. And typically, these different domains correspond to different exons within the gene that the protein is encoded by. I'll repeat that again, but say it a little differently. The exons that make up a gene that encodes for a protein, the different domains of that protein correspond typically to certain exons at the DNA level. And so what ends up happening is during splicing, we've talked about how that splicing typically removes the introns. And so what ends up happening when we have our primary transcript or the pre-mRNA, we have the exons and the introns. If you look at the numbering in the primary transcript or the pre-mRNA, the numbering exists only within the exons and it skips over the intron. So we have nucleotides 1 to 30 in this exon, an intron, and then it picks right back up at 31. And so once we have splicing that removes out the introns, what we have left is this contiguous mature mRNA that goes from the first nucleotide to the 146 nucleotide with a poly A tail on the end. And so again, this is just meant to try and highlight and illustrate that a key takeaway here is that the transcript that's first produced at the end of transcription is a pre-mRNA or a primary transcript. Splicing removes out the introns, and what we have left is a mature mRNA with no introns, contiguous exons present within the mature mRNA with the 5' cap and the poly A tail. Yes. Do we have some exons that are removed? That can happen. So that's how we get alternative splice variants, where there might be one form of this protein where this first exon is gone, or perhaps another one where this larger second exon is gone. And so there's going to be different variants of the same protein, or it will encode for different variants of the same protein. But when it comes to, what's a good way to say this? So I'll break this in terms of like importance. Most important thing to note about splicing is that it removes the introns from the mRNA. Before splicing happens, we have this pre-mRNA, this primary transcript. At the end of splicing, it's a mature mRNA transcript here. Next level is what you just touched on. We can have different exons removed during splicing that can lead to different variants or different versions of the same protein that might have different functions. So because different exons, and this goes back to what we touched on the previous slide, because different exons typically correspond to certain domains, there might be an important domain that's lost in a certain splicing variant. For example, if this was an enzyme that was being encoded for, and this domain encoded for the regulatory domain, we might lose the ability to allosterically regulate that given enzyme. Or perhaps this is actually the catalytic domain. This is the domain where that actually is where the enzyme interacts with its substrate. And so that splice variant where this domain encodes for, that's lost and it goes to the catalytic domain, this might be a enzyme that is rendered non-functioning now. And so there are different consequences or different outcomes that can happen as a result of different splice variants. Excuse me. Right. So what ends up happening is where the splicing occurs, what marks where the introns are within that transcript is actually marked by these, there are consensus sequences called splice sites, which specify the beginning and end of a given intron. They're actually just two base sequences, G and U at the 5' end and AG on the 3' end of that intron, and are 100% conserved across introns. And so they mark these sites where splicing will occur, and they catalyze the formation of what we call the spliceosome, this large multi-protein structure that mediates splicing to happen that removes out that intron. In addition to the spliceosome is made up of a series of proteins and RNAs. We have our what we call SNRPs, our small nuclear ribonucleic particles. And what this looks like is we have our pre-mRNA. I'm actually going to stay at the computer here and try and walk us through with the pointer. So we have our pre-mRNA right here. We have an intron in the middle and an exon to the right and to the left. We have our consensus sequences that mark where splicing occurs. We have the GU sequence on the 5' end of this intron and AG on the 3' end of this intron. What ends up happening is that they mark these, they are splice sites where these SNRPs will come in. The first one is called U1, will bind to the GU 5' end of this intron. We'll have UT not bind at the AG, the far 3' end, but there's actually this what's called a branch point sequence at this adenosine right here. What will end up happening is that after U2 binds, U3, or not U3, sorry, U4, U5, and U6 will come in and bind. And what I should say is I'm not asking you to memorize every step of this process, but rather helping you see and understand how splicing happens. It'll be important to note that there are consensus sequences that mark these splice sites where splicing occurs, but not asking you to memorize the order of events with each component that makes up the spliceosome. And so we have these other SNRPs that come in that form this entire spliceosome here. This branch point sequence becomes really important because we have the disconnection of that 5' end that comes in and binds at that branch point sequence. That'll then lead to the formation of what we call a lariat structure. And event after the formation of the lariat structure, that forms a cue to cleave the 3' end at that splice site, combining the two exons and removing this intron. Because this intron, which is ribonucleic acids, it doesn't have a poly A tail, it doesn't have a 5' cap, it is not protected from degradation and will undergo degradation. And so we've removed this intron that will now be degraded. And exon 1 and exon 2 are joined together, having successfully spliced out that intron that was just degraded. And so this is kind of the big picture description of how splicing occurs, not asking you to memorize each and every single step of the process, but know the main, to understand what enables this process to occur, these consensus sequences that contribute to it, that there are these SNRPs, these small nuclear ribonucleic particles that contribute to the formation of the spliceosome. We're not asking you to memorize every single SNRP that contributes to it and when it comes in. Are there any questions on this slide here? Yeah, yeah.
Or does that mean that like for one protein, one section might be an intron, but it's an exon for another protein?
And that's how we get different splice variants where exons can be removed, where if there is perhaps say a GU and an AG within an exon, that can lead to the removal of some given exon that leads to a specific splice variant. Totally normal in nature, it happens, but there can be instances where that leads to, again, either in the case of an enzyme where it removes the catalytic domain, can render it non-functioning, or that can be a harmful splice variant, or we can just have a smaller version of the same protein that does all of the same things. But you're right. Having GU and AG within an mRNA is going to be present at coding regions within these exons. And so that it's 100% conserved across introns, but can be present in exons that leads to these different splice variants that we've talked about.
So no. Not just random.
How does it know not to randomly?
Of H, no, bind.
Oh, well, so there's also the probability of a GU and an AG being within a given distance that the spliceosome can can traverse that distance. So if there's a GU and an AG that are like incredibly far away, unlikely that the spliceosome will cut out across that wide distance. Typically introns exist within a given range. And so that's where, like, if there's an exon within the same range with a GU and an AG, that's possible. Again, the probability of GU and AG being like some odd distance apart, like that's going to happen, but will be beyond. I'll give it a critical distance. Like beyond a critical distance, splicing is unlikely to occur across that entire mRNA transcript. If that makes sense. Any other questions? Okay, so that marks the end of content. There's two quick announcements. I still have your time. We're not late. Two quick announcements. One is, there we go. There is the opportunity, if you're a rising third year, to take Ideas Lab over the summer. Ideas, for those of you who are still in your second year who haven't taken Ideas Lab yet, it's kind of like the main class you're going to take in your third year. That's a research intensive. It will occupy a lot of your emotional and mental and physical bandwidth as a third year. The ability to take it over the summer, if this was an option that was available to me when I was here, 100% would have taken it. So highly recommend you consider it. There is tonight, I think, yeah, Tuesday the 25th, tonight. There's a Zoom being held tonight with Dr. Allen and a student who I believe has taken it. And so if you're at all interested in making your third year just a little bit easier, you should join that Zoom call and ask any questions and get some more information about that option. Finally, there is the annual BME formal that's taking place on April 5th. I'm going to correct this and say this is actually the 4th annual because there was one when I was a student here. I think it got too rowdy, so they stopped it for like 10 years. I can stay here. But yeah, it's a great time. It's at Random Row this year. There's a QR code for registration, and then at the bottom right, the QR code for nomination for awards for faculty, students, and TAs. It's a great evening. Highly recommend you check it out. With that, I will call it there. Thank you, guys.